Assalamualaikum, I am Dr. Leera, Assistant Professor of Biochemistry, Bashundhara Ad-din Medical College. Today, we are going to learn about RNA, a very important topic of molecular biology. RNA is the full form is ribonucleic acid. What is ribonucleic acid? The polymer of ribonucleotide is known as ribonucleic acid. It is linked together by the phosphodiester bond. There are three types of RNA mainly. These are mRNA means messenger RNA, rRNA means ribosomal RNA and tRNA, that is for transfer RNA. Other specific types are small nuclear RNA and heterogeneous nuclear RNA. RNA consists of three things, mainly the ribose sugar, the phosphate and the nitrogenous bases. The nitrogenous bases that are present in the RNA are adenine, guanine, cytosine and uracil. The RNA, one ribonucleotide unit that is composed of ribose sugar, the phosphate and the nitrogenous bases. One ribonucleotide is connected with another ribonucleotide with the 3' 5' phosphodiester bond. Structure of different types of RNA. First of all, we are going for rRNA or ribosomal RNA. rRNA makes up about 80% of total RNA. It is found in association of different protein as a component of ribosome. There are three distinct size species of rRNA in prokaryotic. These are 23S, 16S and 5S. In eukaryotic, there are four distinct sizes species of rRNA, that is 18S, 28S, 5.8S and 5S. tRNA is very important for you because it comes in the professional examination as a draw and label the picture of a tRNA. So, this tRNA is very important. tRNA makes up 15% of total cellular RNA. It is the smallest of all three major RNAs. Molecular weight of is very less in tRNA. tRNA contains 74 to 94 nucleotide residues. tRNA contains unusual bases. tRNA have extensive interchain base pairing. tRNA is the least one specific type of tRNA molecule for each of 20 amino acid. A tRNA molecule consists of 4 hand or 4 loop. The first is D loop, then anticodon loop, C loop and acceptor arm. We can remember it with this data. D stands for the D loop, A stands for the anticodon loop, T stands for the C loop and X stands for the acceptor arm. In D loop, there is some base pairing in D loop, anticodon loop, C loop and acceptor arm, and this portion is known as non-complementary region. This CCA is non-complementary region. In D loop, there is 4 base pair. In anticodon loop, there is 5. In T loop, C loop, there is 5. In acceptor arm, there is 7 base pairing. There is 4 complementary base pairing in D loop. There is 5 complementary base pairing in anticodon loop. There is 5 complementary base pairing in C loop and there is 7 complementary base pairing in acceptor arm. And this is variable loop. This is the picture of a tRNA. This is the anticodon loop. It's attached with the codon region of mRNA, and this is the acceptor arm for amino acid. The anticodon loop the genetic information from mRNA in this region and accept the amino acid in this region. That's why it is known as the acceptor arm. So, the parts of tRNA molecule are acceptor arm. This arm is capped with a sequence of CCA amino acid attached to acceptor arm. Anticodon loop responsible for recognition of triplet codon of mRNA. D loop. It is named so due to presence of dihydrouridine. T psi C loop. This arm contains a sequence of T pseudouridine and C means cytidine. This is a picture of tRNA. mRNA. mRNA make up 5% of all total RNA in a cell. It is the most heterogeneous type of RNA in terms of size. Special characteristics of a eukaryotic mRNA is composed of 5 parts, the 5' end and the 3' end. The 5' end, there is a 5' cap, and after this 5' cap there is a 5' untranslated region. After that there is a coding region, that is the exons or exons, then the 3' untranslated region, then 3' poly A tail. The coding region, first of all there is a initiation codon, then the exons and then stop codon. Poly A tail. What is poly A tail? A long sequence of adenine nucleotide at 3' end of mRNA chain. A cap. What is 5' A cap? Consisting of 7 methyl guanosine attached backward through triphosphate linkage at 5' end of mRNA. Function. What are the function of these RNAs? rRNA. It acts as a platform where mRNA and tRNA interact for protein synthesis. What is the function of mRNA? mRNA carries genetic information from DNA to ribosome, where it is used as a template for protein synthesis. tRNA. Each tRNA serves as an adapter molecule that contains its specific amino acid to the site of protein synthesis. What is the difference between DNA and RNA? First of all, the difference is in the nitrogenous bases. In DNA, there is adenine, guanine, cytosine and thymine. In RNA, there is adenine, guanine, cytosine and uracil, pentose sugar. In DNA, the pentose sugar is deoxyribose sugar, and in RNA, the pentose sugar is ribose sugar. Location. The DNA is located in nucleus and mitochondria. The RNA is mainly located in cytoplasm. Strand. There is double strand in DNA and single strand in RNA. Types. There is no types of DNA, but RNA has mainly 3 types: mRNA, messenger RNA, ribosomal RNA and transfer RNA. Complementary base pairing rules. Adenine always pairs with thymine and guanine always pairs with cytidine by hydrogen bond, and there is such no base pairing in RNA. Molecular size. DNA is larger than RNA. Mutation. In DNA, mutation occurs and no mutation occurs in RNA. Histone present in DNA, absent in RNA. Denaturation by pH, heat and temperature occurs in DNA, does not occur in RNA. Function. Function of DNA is it is a chemical vehicle of hereditary and fundamental unit of genetic information, and RNA helps in protein synthesis. This is a picture of DNA and mRNA. Thank you for all.